Hello everyone, my name is Alyssa Prater and today is June 4th of 2024. And I'm welcoming you all to my presentation on chemoreception, focusing on the fascinating sense of smell and taste. So let's dive right in. So chemoreception, the sense of smell and taste, in this presentation we'll explore how our bodies detect and interpret chemical stimuli, specifically through olfaction and gustation. Nextly, we will be talking about the introduction to the chemoreception. So, chemoreception is the physiological response of a sense organ to a chemical stimulus. It's essential for detecting changes in our environment, which is crucial for survival, feeding, and communication. Today we'll focus on olfaction, the sense of smell, and gustation, the sense of taste. Olfaction and gustation are vital for our daily lives. They help us detect food, avoid danger, and even communicate. Understanding these senses not only gives us insight into how we interact with the world, but also how our nervous system processes complex information. Next, we will be talking about the sense of smell, also known as olfaction. Olfaction begins in the nasal cavity, where odor molecules are detected by the olfactory receptors located in the olfactory epithelium. These receptors are specialized neurons that can bind to specific odor molecules. Once an odor molecule binds to a receptor, it activates a signal transduction pathway. The pathway involves G protein-coupled receptors, which trigger a cascade of intracellular events. These events lead to the generation of an electrical signal, which is transmitted to the olfactory bulb via olfactory nerve. The olfactory bulb is a critical structure located at the base of the brain. It processes the signals received from the olfactory receptors and organizes them into a pattern that the brain can interpret. The information is then sent to the olfactory cortex, where it is further processed, leading to the perception of smell. The complexity of the olfactory system allows us to detect and distinguish between thousands of different odors. This ability is essential for identifying food, scents, sensing danger, and even recognizing individuals based on their unique scent. Next, we will be talking about the sense of taste, also known as gustation. So similar to smell, gustation involves detecting chemical stimuli, in this case, taste molecules. Taste buds located on the tongue and other areas contain receptors that can identify 5 basic tastes: sweet, sour, salty, bitter, and umami. Taste buds are composed of gustatory cells, which are sensory receptors for taste. Each gustatory cell has microvilli, known as taste hairs, that extend into the taste pores. These taste hairs interact with the molecules from the food that we eat. When a taste molecule binds to a receptor on the gustatory cell, it activates a signal transduction pathway. Depending on the type of taste, different mechanisms are involved. For example, salty and sour tastes primarily involve ion channels, whereas sweet, bittery, and umami tastes involve G protein-coupled receptors. Once a signal is generated, it is transmitted to the brain via cranial nerves, including the facial nerve, glossopharyngeal nerve, and vagus nerve. These nerves carry the signal to the brainstem, where initial processing occurs, and then the gustatory cortex for further interpretation. The perception of taste is not just limited to the tongue. It also involves the palate, pharynx, and even the epiglottis. Together, these areas contribute to the overall experience of taste. Next, we will be talking about signal transduction and chemoreception. Signal transduction and chemoreception involves converting chemical signals into neural impulses. This process begins when a chemical stimuli binds to a receptor, triggering a series of intracellular events. In olfaction, odor molecules bind to G protein-coupled receptors on the olfactory receptor neurons. This binding activates a G protein inside the cell, which then activates an enzyme called andylate chylase. Andylate chylase converts ATP to cAMP, which opens ion channels, allowing ions to flow into the cell and generate electrical signals. In gustation, the mechanisms vary depending on the type of taste. For salty and sour tastes, ion channels play a significant role. Sodium ions for salty or hydron ions for sour either enter the gustatory cells through these channels, directly causing depolarization and generating a signal. For sweet, bitter, and umami taste, G protein-coupled receptors are involved Binding of the taste molecules to the receptor activates a G protein, which then activates a second messenger system inside the cell. This leads to opening of the ion channels and the generation of an electrical signal. These signals are then transmitted to the brain, where they are processed and interpreted, allowing us to perceive different smells and tastes. Next, we'll be talking about the integration of smell and taste. So smell and taste work together to create the perception of flavor. This integration is crucial for enjoying foods and drinks. The combined pathways of these senses in the brain enhance our overall sensory experience. The integration occurs primarily in the brain, where signals from both the olfactory and gustatory systems converge. The brain combines these signals to create a unified perception of flavor. The process is which allows us to experience the richness and complexity of flavors in our food. For example, when we eat an apple, the sweetness we perceive is a combination of the taste detected by our gustatory system and the aromatic compounds detected by our olfactory system. This integration enhances the overall experience, making it more enjoyable and memorable. Moreover, the combined sensory experience is essential for detecting the quality and safety of food. If something smells or tastes off, it's an indication that the food may be spoiled or contaminated, helping us to avoid potential harm. Next, we'll be talking about the application and importance of chemoreception. Chemoreception is vital for detecting hazards like spoiled food or dangerous chemicals. It also plays a significant role in nutrition and dietary choices. Moreover, advances in technology have led to the development of artificial noses and taste sensors, which have various applications in medicine and environmental monitoring. Artificial noses, also known as electronic noses, are devices designed to detect and identify odors. These devices mimic the human olfactory system and are used in various industries, including food and beverage, environmental monitoring, and healthcare. In the food industry, electronic noses are used to ensure the quality and safety of products. They can detect spoilage, contamination, and even subtle differences in flavor profiles, helping manufacturers maintain high standards. In healthcare, artificial noses are being developed to detect diseases based on changes in body odor. For example, certain diseases can cause characteristic changes in a person's breath, sweat, or urine. By detecting these changes, electronic noses can aid in early diagnosis and monitoring of diseases. Taste sensors, also known as electronic tongues, are used to analyze the taste profile of substances. These devices are particularly useful in the pharmaceutical industry for improving the taste of medications, making them more available for patients and making patients feel less sick when having to take a medication. Overall, the advancements in chemoreceptive technologies have the potential to revolutionize various fields, improving quality control, disease detection, and environmental monitoring. Next is the conclusion. So to summarize, chemoreception involves the sense of smell and taste, which plays crucial roles in detecting and interpreting chemical stimuli. Understanding these processes helps us appreciate the complexity and importance of the senses in our daily lives. We've explored the mechanisms of olfactory and gustation, the pathways involved in signal transduction, and the integration of these senses to create flavor. We've also discussed the practical applications and importance of chemoreception in various fields. Thank you so much for watching, and if you have any questions, feel free to comment or leave them below. Again, my name is Alyssa Prater, and today is June 6th of 2024. I hope that you have a great day, and thank you for joining me on this journey through chemoreception.